do we have to care about solutions we put in our bloodstream versus what our plasma is? That's because this thing called tonicity. You've already seen our 5% dextrose and our saline are at 300 milliosmoles. So we've already talked about isotonic, meaning the same. However, we've got two other terms, hyper and hypotonic. I think we can figure out hyper, right? Hyper is, what does that mean? Just the prefix hyper. High, too much. If you can't appreciate that one, babysit a hyperactive kid. You will figure it out and never, ever forget. So a hypertonic solution is a solution that has a high amount of what? What's our fancy S word? Solutes. Hyper refers to solutes. Versus hypo, which means low or below, is a solution that has fewer or a lower concentration of solutes when compared to the other side of a semi-permeable membrane. For our example, we're going to use red blood cells because you're injecting saline, dextrose, any other kind of solution into the plasma, that's going to bathe your red blood cells. So if you shift the balance of solutes, you can have an impact on the water concentration inside those red blood cells. So here's a nice illustration. I like pictures. Is anybody else a visual learner? Pictures? I like pictures, but I also have the text for my more English literary readers to go through. And then I guess the videos are going to be for, I don't know, whoever likes those. Okay. So here's our animal cell, isotonic solution. You have our nice biconcave red blood cell. So in an isotonic solution, what does the water do? Be very careful, it's kind of tricky. What does the water do to a red blood cell in an isotonic solution? Since the red blood cell stays the same shape, you might be inclined to say the water doesn't move. But in fact, it does. It's just an equal amount of water moves in as it moves out. So the net osmosis in an isotonic solution is zero. It's neutral. But if you drop a red blood cell into pure water, that reverse osmosis water that we made earlier this morning or this afternoon, if you drop that red blood cell into a solution of water with no solutes, where's the highest concentration of solutes? In the red blood cell or out? Inside, right? So now the proteins, the nucleic acids, the glucose, the stuff inside that cell is going to be that water magnet. And it's going to draw the water into that red blood cell. So more water is going to enter than could leave. And so that red blood cell is going to swell up until it ruptures, until it bursts. Big fancy word is lysing. Have you ever overfilled a water balloon? Yeah, that's what we're doing. Now conversely, if we put that red blood cell into seawater, the salt in that water is a higher concentration of solute than inside the red blood cell. So now where are we going to see the net movement of water? In or out? That salt water is going to just suck the water out of that red blood cell because that osmotic pressure, the drawing force of the solutes, more water leaves than enters and the red blood cell shrivels up. What's our big fancy name for that? Crenate. Red blood cells are going to be crenated. Neither a hypotonic or hypertonic solution is going to be a happy solution for our red blood cells. So that's why that is so super important to make sure the solutions you introduce into the body are isotonic. Everything you wanted to know about diffusion, but just really never thought of, right? All right, changing chapters now. We go to carrier mediated. For whatever reason, these molecules may be too big, too charged, cannot cross the phospholipid membrane without the assistance of a protein carrier, a channel, a pump. And so the first one we're going to talk about is facilitated diffusion. Now, this is carrier mediated. We use a protein. But since it's diffusion, how much energy is going to be used? None. Zero. What is the direction in which the molecules are going to move? High to low, still downhill. Exactly like diffusion. The only difference is you've got to go through a protein. You've got to go through a protein. You simply can't diffuse through the plasma membrane. Now, some of these are going to be really important in the fact that they are just not tunnels. They're simply not culverts. But they're going to have gates built into them so that you can regulate when diffusion can happen and when it can't. You see the advantage? You add in a level of complexity. These molecules can't just diffuse all the time. You control when they diffuse. Just like the hydroelectric dam, you don't necessarily generate electricity all the time. If the water's low, you don't generate as much electricity. If the water level gets really high, you have to open up the floodgates and everything. So some of these carriers are going to be gated. We're going to see those in our neurons, and they're extremely important. That's going to be in the next chapter. So some are gated, some do look like these passageways, but others are actually going to be able to change shape. And even though this protein is changing shape, in our example here, how do we know this is facilitated diffusion and not active transport? Look at our illustration. What do we have inside? High concentration in, and the molecules are moving to a low concentration. That's diffusion. Even though our carrier is moving, is changing shape, is actually transporting, it's still diffusion. The potential energy of the gradient is what's driving that level of transport. Now, active transport. This is going to be the one that is most different. We're still going to use a protein. It's still carrier mediated. But now everything else is reversed. Do we use energy for active transport? Yes, that's why we call it active. And active transport is going to be the first and possibly the only transport mechanism where we're transporting materials uphill or upstream against the concentration gradient. So those are the big differences. Now, let me go ahead and give you a little preview here. My exams are going to be some fill in the blank. They're going to be some multiple choice. And they're going to be some short answer. My classes in the past have called them baby essays. I don't know. You're going to have maybe 3/4 of a page. That's all I need. I don't need a whole book on this stuff. But one of my favorite questions to ask for the short answer are compare and contrast. You know the difference between those? When you compare, tell me how they're the same. And when you contrast, tell me how they're different. So if I ask you to compare and contrast facilitated diffusion versus active transport, well, they're the same because they're transport, they use proteins. They're different in the use of energy versus the direction of transport. You see what I'm saying? So that's just an example of what that's going to be like, just so you know. Now, I'm also throwing in a new term here that you may not have heard before. We've heard of active transport, right? Have you ever heard of primary active transport? We're going to distinguish it at another level of complexity. Primary active transport is the movement that occurs from the direct utilization of energy. And what form of energy do we have in all of our cells? ATP, adenosine triphosphate. It's that linkage between the second, third phosphate. That's our energy that we can use. And so the direct work that is accomplished from the utilization of that energy is called primary active transport. Now we're going to have 5 steps. I think I might only list 4, but we're going to break that last one down into 5 steps. So when we look at primary active transport, we're going to bind the transport molecule to the carrier. It's got to get stuck there first. If it doesn't get stuck, nothing's going to happen. The second thing that happens is that carrier protein is going to have the ability to bind with ATP and convert it into ADP and an inorganic phosphate. It releases the energy of that chemical bond. We say that that is ATPase activity. Anytime you hear ASE on the end of a word, that means it's enzymatic. So our carrier has ATPase ability, and it can release and capture that energy. So think of the carrier molecule bindings like putting the key in your car. With ATP present and that conversion, that's like starting the engine. And because of that energy being released, we are going to get a conformational change in the carrier. That protein changes shape. And as that protein changes shape, it's going to move that molecule, that ion, into or out of the cell. Now, this is where we're going to break this down into 2 steps. So letter D, once you have that conformational change, then the ion, the transport molecule, gets released into the next compartment, either inside or outside, depending on the direction of movement. Letter E or number 5, however you want to do that, the last step, the carrier protein resets itself. It returns to its starting configuration to do it all over again. Now, do you need more energy to reset? No, it's like an elastic band. That's the rebound. So that all of that is our primary active transport. We just moved an ion from one place to the next. So here we've got a little animation showing the binding of the molecule, the release of the energy, the resetting, and we're doing it again and again and again. There's one thing about this illustration that I hate. My OCD just jumps up and it grabs me by the throat and it says, find a different illustration because it's wrong. What's wrong about our illustration here for active transport? The concentration gradient. You should have a whole bunch of red dots in here and just a few moving in. Be very careful and be critical when you read science, when you look at illustrations on the internet. Well, if it's on the internet, it's got to be true, right? No. Or even in class when you get presented materials. Because artists are putting this stuff together, not necessarily the scientist. I like it because it moves, and again, I'm a visual learner, but you see the concentration gradient is not as it should be for active transport. But how do we know this illustration was for active transport? What's being used? Energy. Energy is being used, so we know, okay, well, they were trying to illustrate it, they just left off the high concentration of red dots inside. Okay? Active transport. Now, we're going to look at an example of active transport. Our example is going to be our sodium-potassium pump. We are going to use these everywhere in the body. We're especially going to use these in the kidneys to help us conserve water. We're going to use these in our neurons so that we can have more than one nerve impulse a day. And in fact, there's so many sodium-potassium pumps in your body, active transport pumps that use energy. 12% of the ATP that's used in your body every day is used by the sodium-potassium pumps. That's a ton. So we're going to learn this, and we're going to learn it very, very well. We talked about concentration gradients of ions inside versus outside the cell. Where is sodium at the highest concentration relative to the cell? Outside. Now, here's a little trick. Sodium and potassium are almost always opposite. So if you learn that sodium is highest outside, you automatically know where's potassium highest? Inside. Don't you love learning half as much as you need to know, but you know it? See, that's part of my job is not just to help you learn it, but help you be able to remember the stuff. So sodium is high outside. Part of the reason that sodium is in high concentration outside the cell is because of the sodium-potassium pump. So to keep that high sodium concentration, guess what the sodium-potassium pump is going to do? It's going to move 3 sodium ions where? Outside, to keep the sodium concentration high. To keep it as high as we can, because when sodium's going to diffuse, we want it to go as fast as it can. And what's one way we increase the rate of diffusion? Make the hill steeper, make the gradient higher. Now that is primary active transport. When the sodium-potassium pump binds 3 molecules of sodium inside, it cleaves the ATP. It has a conformational change, and it releases those 3 sodium outside the cell. That's primary active transport. But the sodium-potassium pump does something really tricky next. When it releases those 3 sodium outside the cell, it gets stuck. It gets locked in that configuration. And it can't reset yet. To be able to reset so it can move 3 more sodium ions by using another ATP molecule, it has to bind 2 potassium ions outside, which act like the keys to unlock it, and then it resets. And as it resets, it pulls those 2 potassium ions in and releases them inside the cell, and that's its final resetting, reconfiguration, so it can do it all over again. Now, how much extra energy did we need to use to move those potassium ions? Did we have to cleave another ATP molecule? No, you're going to reset anyway. Why don't you just take me with you? And so that's how we distinguish primary from secondary active transport. The direct effect of the expenditure of energy, a secondary consequence that happened in spite of the fact that we didn't have to use more energy. How many of you have ever experienced the joy of someone saying, "Hey, I heard you're going to Dallas this weekend. Can I catch a ride?" And then you ask them to pay for gas. It's like, well, you were going anyway. Has this ever happened to anybody? Right. You're the primary active transporter. The person who wants a free ride would be the secondary active transporter. Now, the fact that we're moving ions in opposite directions by the same carrier, we can refer to the sodium-potassium pump as an antiport carrier. Anti meaning in opposite directions. And so here is our illustration. Again, they don't have the concentrations right, but we'll kind of ignore that. Artistic license. But it does show you the binding of the sodium, the utilization of the ATP, sodium going out. Then to be able to reset, we have to bind the 2 potassium so that we can get back in and do it all over again. Any questions about primary versus secondary sodium-potassium pump? Got it? Okay. Now again, I've already mentioned these pumps are crucial for maintaining the concentration gradients. Here you can see we've got a bunch of our sodium-potassium pumps working in our plasma membrane. We can see inside the cell, intracellular, outside the cell, extracellular. Different books, different illustrations are going to offer you different numbers. I'm not so hung up on the exact number. Some places we will. But here, I want you to know that we're around 150 millimolar outside for sodium and around 150 millimolar inside for potassium. Look at this, 150 versus 5, 145 versus 12. That's almost a straight skydive, right? That's about as steep as you can expect, which means when we allow diffusion to occur, it's going to be like jumping out of an airplane. It's going to be really, really fast. Now, we've already seen antiport with our sodium-potassium pump. Two ions moved in opposite directions by the same pump. We can have a symporter, which moves molecules, two different ones in the same direction, or we can have a uniport, one transport, one direction. So when it gets to test time, it's going to be critical, absolutely critical, that you read the question carefully. So for sodium-potassium pump, you know, I could ask you a question about what kind of transport mechanism would you call the sodium-potassium pump? If I asked a question like that, what type of transporter is the sodium-potassium pump? What are some possible answers? Antiport, that would be correct. Active transport, that would also be correct. What else? It'd be the antiport for the pump. If it was the same direction, it'd be symport, and we're going to get to one of those. You could also say it's carrier mediated. All three of those would be correct. So I have to define the term that I'm wanting to see if you know. So for instance, what term could you apply to the sodium-potassium pump that refers to the carrier transporting two different molecules in opposite directions? Active transport wouldn't work there, right? So the question is going to be critical. When I find students that don't do well on exams and they come into my office to check their tests, how many times have you done that? You go in, I missed that. Oh, no, I know that one. Oh, why did I put that? I didn't read the question, right? Read the questions carefully. So this next example of transport is going to further separate primary and secondary active transport in a very profound way. And we're going to use epithelial cells like in your intestines. We've already talked about the intestines, absorption of materials at increased surface area. And when we look at our epithelial cells, we're going to see that we have a symport pump in the apical or luminal surface of your intestinal cells. These are the ones that have the microvilli on them. And this symporter is going to be a glucose sodium symporter. Okay, same direction. So let's take a look at this. Here we've got that Big Mac we've just eaten. It's been processed, broken down. The glucose has been liberated. Now we need to get the glucose, which is low concentration, outside the cell. We need to get it inside that cell. Now, we say it's low concentration outside simply because that lumen is so big, it's so dilute. All right? We haven't concentrated it in our bloodstream yet. But it's important we get every last bit of glucose into the cell. And so the way we're going to do that is we're going to couple it with sodium. Sodium, high concentration outside, low concentration inside in this case, right? Because we know sodium's high outside. And so glucose sees that sodium's going to Dallas, and what does glucose say? Hey, can I catch a ride? Can I go with you? And so as sodium's coming in, it brings glucose in with it. Now, glucose is low outside, high on the inside. We're going from a low concentration to high concentration. What does that sound like? What's our only low to high? Active transport. So do we use energy here? That's a trick one. Be careful. No, good answer. No, we don't use energy. Not directly. So this is an example of secondary active transport. Why is it secondary active transport? How did we create our low sodium concentration inside the cell? What's our friend we just talked about? 3 sodium out, 2 potassium in. Sodium-potassium pump. It uses energy to move sodium out, primary active transport. But since we created that low concentration inside, we take advantage of it to allow the sodium to diffuse in, and as it's diffusing in, here comes glucose with it. Carrier mediated, facilitated diffusion, and glucose just catches a ride. So now, man, we've got all kind of terms we can apply to this one, right? Symport, facilitated diffusion, secondary active transport. Read the question. My job is to direct you. It's not to trick you into seeing if you can figure. Have you ever been in those classes? You read the questions on the test and you don't have a clue what it is they're asking you for. This stuff is complicated enough. I just want to see if you know it, and you've got to be able to understand the question to be able to formulate an answer. So when we look at all this, we're going to see that sugars, like we saw with glucose, some amino acids are going to be transported in this facilitated way. And again, we've got that facilitated diffusion as a secondary active transport mechanism with the sodium-potassium pump providing the potential energy in the form of that gradient through primary active transport. So here's the rest of the picture. Here you can see what's happening at the apical surface, but the reason sodium can diffuse in is because of the active transport that's occurring at the basal surface to transport the sodium out of the cell and into the bloodstream. And then once we get glucose into the cell, then guess what? Glucose is higher concentration in here than in the blood. We get a uniport. So what kind of transport would be happening here with the glucose? It's from high to low through a protein. Not just diffusion, you got to put something in front of it. Facilitated diffusion. When you say just diffusion, it's like the one before the A in algebra. It's implied that diffusion means simple diffusion. To designate facilitated, you've got to tag the facilitated in front of it. So here we've sort of put all those transport mechanisms together in this one cell. Hopefully you're beginning to appreciate the complexity of cells, and as we get bigger and put in the organ system, how complex the human body is. It really is an intricate machine. Now with our concentration gradients, sodium highest where? Outside. Potassium, you could say opposite or inside. All right? I've repeated that a couple of times, right? When a prof repeats stuff, what does that mean? It's important. I had a, what? I always got a student says on the test, which means it's also important. Right. Sodium high outside, potassium high inside. Now we're going to turn our attention to the charge of the ions. We're going to create a battery. We're going to create a biological battery. You got a positive side and a negative side. And when you have that charge difference, you set up a voltage, a potential. And we're going to call this our membrane potential. And a couple of things that are going to be important: concentration of gradient, which we've already seen with our sodium and potassium, and those are the ones we're primarily working with. But also the permeability of those ions, the ability of these ions to pass through their respective protein channels. Some of these ion protein channels are going to be locked tight, but other protein channels are going to be kind of leaky. And they're leaky on purpose. And so when we look at permeability, we're going to see the ion that makes the biggest difference with our membrane potentials is going to be potassium. All right? Now, here's another one that doesn't necessarily and routinely come to your mind, because we have a lot of anions. And what charge do anions have? Negative charge. What do we call the positive charged ions? Cations. So we have a lot of anions that are said to be fixed. They're ions inside the cell that are tied up in nucleic acids, in proteins, in amino acids. These negatively charged molecules can't get out of the cell. And these fixed anions are principally why the inside of the cell is going to be more negative than the outside of the cell when the cell is at rest. These fixed anions, these negative charges that can't go anywhere, are principally what causes the inside of the cell to be more negative at rest than the outside. Now, once again, as I mentioned before, potassium. Of the two, potassium and sodium, potassium's the most permeable. So it's going to be the one that's going to be the most influential of what our membrane potential, that charge difference, that voltage across that membrane. Potassium's the one that's going to play the biggest role in determining that, simply because it's kind of leaky. And again, there's our charge difference inside 150 versus 5. Here we've changed a little bit. We're not talking about molal anymore or molar. We're talking about milliequivalents, and milliequivalents just takes into account our valence, or the charge of a particular ion. But notice the numbers didn't really change. So our milliequivalent still for potassium, we're still high on the inside, low on the outside. Sodium, just the opposite. High outside with about 150 out, 12 in. But the difference here is sodium is not as permeable. It's not going to play such a huge difference in what our membrane potential is at rest. Okay, so here's my big picture. Let me explain this a little bit. I like pictures that have a ton of information in them, because I could take that snapshot and kind of think of them again. They're trying to show you the phospholipids. You see them here? There's our charge head, our two fatty acid chains. So there's our phospholipid layer. Who do we have right here in the middle? What is that? 3 sodium, 2 potassium. Sodium, potassium pump. Here we're using our energy. This is inside the cell. This is outside. So we're moving our 3 sodium out, our 2 potassium in. So this shows you our gradient. This is going to be for potassium. Here's our gradient for sodium. Sodium high out, potassium high in. So that puts a lot of meat, a lot of information in this one picture of what we've been talking about a lot today already. The one thing that, this is messing with me now. The one thing that I wish that I could add to this is to somehow, and maybe you can, if you've got the slides, maybe for this arrow, just put like 1 with a degree for primary, and for this arrow, 2 with a degree for secondary. Right? That's our primary versus secondary active transport. I think that's the only other thing that I would like to put in there to help make it even busier. I don't know, more information. Now, this, above all else, is a biology class. You will not need a calculator on my exams. But you are going to need to know the equations that are used and how to interpret the product of these equations and what it means. Okay? So when we look at our equilibria, our voltages, our valences, our membrane potentials, there's something that's called an equilibrium potential. And it's calculated by an equation called the Nernst equation. Doesn't that look horrible? I know there's worse math out there, but I don't like math. So the Nernst equation for potassium takes into account its concentration gradient. And in doing so, we come up with an equilibrium potential that says at minus 90 millivolts, there will be an equal number of potassium ions inside versus outside the cell. That's where you get a net movement of 0 of potassium when it's diffusing at a membrane potential of minus 90. For sodium, that potential is going to be positive 66. So once again, we're looking at the inside of the cell. And when we have a membrane potential that is negative, that means the inside of the cell is more negative than the outside. Which ion did we say influences that potential the most? Which one has the leaky channels? Potassium. That's another reason why the inside of the cell is more negative when the cell is at rest. The fixed anions and the leaky potassium. But it's not just potassium and sodium. We take into account all of the ions, all of the fixed, all of the movable, transportable ions, and that's where we arrive at what we refer to as our resting membrane potential. All these concentrations, all these charges, the effects and the work of the sodium-potassium pump. Here, once again, potassium high inside, sodium high outside. We've also got calcium, which is highest outside the cell. And then we've got chloride. Remember, chloride's always going to follow sodium, so chloride's going to be high outside. All of these collectively adding together are going to have us arrive at our resting membrane potentials. And the number that I want you to remember for our resting membrane potential is this number here at the bottom, minus 70 millivolts. Now for different cells and even different neurons, there's going to be a range of these. To simplify what we're doing, I want you to remember minus 70 millivolts. Like I said, there's not a lot of numbers I want you to commit to memory, but resting membrane potential of minus 70 is one of them.